Biochemistry of amino acids. You are watching my YouTube channel, Dr. Biochem Lectures. My name is Dr. Alamzeb. I am a professor at the Department of Biochemistry, University of Malacca. I did PhD from the Institute of Biochemistry, Graz, University of Technology, Graz, Austria. This video lecture will include introduction to protein, amino acids, classification of amino acids, and reaction of amino acids. First, proteins. Proteins are a major constituent of most of the cells, more than 50% of the dry weight. Proteins are extremely sophisticated molecules or multi-molecular complexes. An extremely large number of protein types exist in nature. All protein consist of polymer of amino acid that are folded into specific conformation. Amino acids. Proteins are made of 20 different types of amino acid monomers. Amino acids are organic acids containing an amine group and carboxylic acid group. In this structure, you can see that amino acid carbon contain amino group and carboxylic acid group and R group. R group may be an organic residue. Amino and carboxylic acid group remain the same, while only R group are usually different. The most common amino acids are alpha-amino acid in nature. Only 20 L-amino acids are used to make proteins. In nature, only L-amino acids are present. You can see here, alpha-amino acid is the type of amino acid in which amino group is found one carbon away from carboxylic acid, while the beta-amino acid are those which are attached to the beta carbon of the chain. Here are examples of the alpha-alanine and the beta-alanine. The alpha-alanine, in case of alpha-alanine, amine group is attached to alpha carbon, while in the case of the beta-alanine, the amino group is present at the beta carbon atom. Rare exceptions are bacterial membrane proteins, which contain a few D-amino acids, which are usually modified amino acids in nature, primarily lysine and proline, and occasional incorporation of the rare amino acids such as selenocysteine also exist. Classification of amino acids. It is important to know that the amino acids are classified based on the R group properties. The first class of amino acid is hydrophobic or non-polar amino acid. The second class is known as polar amino acid. The third class is known as aromatic amino acids. Now, the first class, hydrophobic amino acid. Hydrophobic amino acids are also known as non-polar amino acid. In this picture, you can see the example of non-polar acid include glycine, alanine, proline, valine, leucine, isoleucine, and methionine. The shaded part, which is the R group, are the non-polar aliphatic hydrocarbon chains, which may contain sulfur or maybe a simple aliphatic chain, or maybe a ring chain, like in the case of proline. The second class of amino acid is known as polar amino acid. Polar amino acid is, or basically three main types. The first type is polar uncharged amino acid. Then the second one is polar positively charged, and then polar negatively charged. So it means uncharged, positively charged, and negatively charged. The first class of the polar amino acid is polar uncharged amino acid. In this case, the R group is usually non-polar, so no net charge is present. Example are serine, threonine, cysteine, asparagine, and glutamine. In this case, you can see here either like serine and threonine, these are hydroxyl containing, while cysteine is CH sulfur containing amino acid. Second class is positively charged amino acid. This class contains net positive charge on amino acid, and the net positive charge is due to the presence of one or more extra amino groups. You can see here in the case of lysine, arginine, and histidine. In lysine, you have one extra amino acid, while in the case of arginine, we have two extra amino group. Similar is the case of the histidine, where we have two extra amino groups. So the amino group increase the positive charge. Now the third class is negatively charged amino acid. In this case, the net negative charge is on the amino acid is because of the extra carboxylic acid group. We have two important examples. The first one is aspartate, the second one is glutamate. Third class of amino acid is aromatic amino acid. Aromatic amino acid contain aromatic ring or benzene ring. These three are the example of the aromatic amino acid: phenylalanine, tyrosine, and tryptophan. You can see in the first example, phenylalanine have only benzene ring, and then the other have hydroxyl group, and then a little bit more modified. Now reactions of amino acid. Amino acid reactions are usually based on either carboxylic acid group, or they are because of the amino acid group, or they may be because of the double bond present in aliphatic chain, or only aliphatic chain. So the reactions of amino acid are based upon these three properties. First reaction is, which is a color reaction, is known as Ninhydrin reaction. In this reaction, Ninhydrin react with the amino acid forming purple color. Ninhydrin normally react with the pre-amino alpha-amino acid group. This reaction is usually used for qualitative and quantitative analysis. Ninhydrin react with the amino acid. With the loss of water molecule, it forms a bond, and then with the addition of another one, it forms a complex molecule with the purple color. Second reaction is the Millon test. This test is specific for tyrosine. If the protein contain tyrosine, the phenol group of the tyrosine is first nitrated by nitric acid. Then the tyrosine complex is mercury ion in the solution to form a brick red. Appearance of red color is positive test for the presence of alanine. This is the mechanism of the reaction, in which tyrosine react with the nitric acid, and then the presence of mercury, a complex is formed, which is red in color. Third reaction is Xanthoprotic test or Xanthoprotic reaction. Concentrated nitric acid react with aromatic nucleus present in amino acid side chain, usually call it nitration reaction. This gives the solution yellow color, You can see here tyrosine react to form this special compound. Similarly, tryptophan also react, but the phenyl, remember that phenylalanine do not react with the nitric acid, and this is because of the presence of the groups on the phenylalanine. Fourth is lead sulfide test or lead sulfide reaction. This test is specific for acid containing amino acid. Sulfur is converted to sodium sulfide by boiling with 40% sodium hydroxide, and then sodium sulfide can be precipitated as a lead sulfide from an alkali solution. When adding lead acetate, we obtain the specific blue color. So cysteine in the presence of sodium hydroxide react in forming sodium sulfide, and then serine, and this sodium sulfide then when reacted with lead acetate it forms lead sulfide, which is the indicator for the sulfide reaction. Fifth reaction is decarboxylation reaction. This decarboxylation reaction can also be carried out in the lab, but this one is the one we are talking about is usually enzymatic reaction. In this case, loss of carboxylic acid group occur. Histamine is produced. The important example is histidine is converted to histamine, which is a neurotransmitter. In the presence of histamine, decarboxylase enzyme. Next is oxidative deamination. In this case, the amino group is removed. You can see here glutamate is converted to alpha-ketoglutarate in the presence of glutamate dehydrogenase enzyme. The next reaction is transamination reaction. Amino group is transferred to a keto acid to form another amino acid. This is very common reaction. We call it aminotransferase reaction or transaminase reaction, in which alanine react with alpha-ketoglutarate forming pyruvate and glutamate. This is an important reaction. Now, an important physical property of the amino acid is absorption of UV light. Amino acid usually absorb UV light, and absorption, for example, absorption of tryptophan is 4 times higher than tyrosine under similar condition. You can see here the absorption spectra of tyrosine and tryptophan. So tryptophan absorb more light than the tyrosine, and this reaction is used. This property of the amino acid is used for the qualitative as well as quantitative determination of amino acid.